Hello student, today I am going to teach you DNA replication that I have given in last few days before. And first of all we have to understand what is replication. It is given here. So replication is the process of producing two identical copies of DNA from original DNA. Okay, that means here you can see this is the model of DNA, and the black color indicates the original DNA, and from this original DNA two new identical copies are producing here from one strand. This new strand is producing, and from other strand these fragmented DNAs are producing. And this process of producing new copies of DNA from the old one, this black color indicates the old one, called template. This process is called replication. In simple term we can call, let me show you one thing. We can compare the process with the Xerox machine. Let me show you one Xerox machine. Suppose here this is the machine in which we insert a paper, and another thing is ink is required, and thirdly electricity is required. Ultimately from the original document, the new copies will be produced later on. And for this what we need, ink is required, and the source of electricity, and the machine itself, and the original document. That means whatever we have to Xerox. In the same manner, in DNA replication also, suppose this is DNA, original DNA, and one end of DNA is 5 prime, that we have understood in class 11 also, and other end is having 3 prime to 5 prime. This is known as polarity of DNA, and these two are the sugar phosphate backbone. And from here you can see these are the nitrogenous bases which are joining to sugar phosphate backbone, and we call ATGC. Suppose this is A and this will be T, and suppose this is C, this will be G, and suppose this will be C, this will be G, and always pairing will be there between C with the G by forming triple bond. Here I have not shown the triple bond, just showing the cartoon model, and T pairs with A by forming double bond between them, and these are the hydrogen bond that we have already understood in class 11 also. Here this is the original DNA, we should call template, and in order to produce the new strand from this old, the new process will be there. So let me show you one thing, that these two bonds are actually joined with each other tightly. This is a hydrogen bond. To make a new DNA from this old one, first of all this bond has to be broken down. Let me show you how it can be broken. So this is a long strand of DNA. It is very difficult for the cell to copy new strand of DNA. For that, from one end or from other end, DNA will open, and it will not open randomly. There must be some point from where DNA will be open and replication starts actually. That point is called origin of replication. For that, please refer to our textbook. Here it is written origin of replication. Here it is origin of replication. Please note that this you must go through these points thoroughly first, and then you'll understand. So let's come back. Now let me draw the DNA again. This is the original DNA, and here again the bonds are forming in this way, bonds join to strand. These are the ATGC. So in order to make a new DNA from this old, that is template, first of all the small stretch of DNA has to be open in this way, and it looks like a fork, and this is called replication fork. So how it happens actually? So first of all we should know the polarity. This is suppose 3 prime, and this will be 5 prime end, and this will be automatically 3 prime, and this end will be 5 prime. This is the original DNA. So now we are in a cell. So let me show you one cell. In a cell there are chromosomes in nucleus. These are the chromosomes. You have seen in class 11 also. This is centromere. In chromosomes we have DNA. Okay. During cell cycle DNA will be produced. DNA will be produced from these chromosomes. So actually chromosome will be duplicated. So after the replication of DNA, what will happen? You see, the cell will synthesize DNA this way. These are the chromosomes. So originally there were 2 chromosomes, and later on this chromosome is divided into 2, and again this chromosome is divided into 2. At last I am going insert. At last what will happen? 2 cells will be produced. So in one cell there will be 1 chromosome, and in another cell there will be another chromosomes. So again that is what I am saying. This chromosome is divided into 2, and at last they are separated to these 2 cells. Similarly, this chromosome is divided into 2, and they will also go to the respective cell in this way. So during this division, DNA is also dividing, and this DNA, originally there was one DNA in this chromosome, another DNA in this chromosome. So this one DNA will be duplicated and replicated, or duplicated we can say, and it will form 2 copies. Similarly, here also this process goes in the same manner, and here we have 2 copies of DNA. Okay, so 1 copy will go to this cell, another copy will go to this cell. Similarly, from another chromosome, 1 copy of DNA will come here, and another copy will go to this cell. So this process is going on in cell. So let us forget about the chromosome, come to the DNA part only. We are in the chromosome. Actually DNA is there in chromosome. How it happens actually? Let's see. Here are the bonds actually, and this bond has to be broken. What I have already explained. These bonds are broken. In above case you have seen bonds are not broken here, but here bonds are broken. How it happened? From one end an enzyme will move that I have already given in the last class, that name of enzyme is topoisomerase and the helicase. These two enzymes will move, and helicase enzyme will break this bond. Okay, so helicase enzyme starts moving from this end, and it goes continuously by breaking this hydrogen bond, and now it has reached this point. Okay, it has reached this point. Topoisomerase is another enzyme. You see, topoisomerase. This enzyme is also moving just above this helicase, and suppose it has reached this point. Topoisomerase has another role, that means it cuts one of the strands somewhere, and it will make DNA replication easy. Later on, this DNA will be joined by enzyme ligase. Here is ligase. So no need to concentrate on the role of topoisomerase, as it will be explained in the higher classes. Now, this is the original DNA. This is called template. Now we are in the cell. Actually, we are in the cell. In a cell, suppose let us go to the first cell. In the cell, we have this A, T, G, C, which are distributed randomly here in the nucleus itself, and these are A, T, G, C are not singly occur. They are already bound with the phosphate and the sugar, pentose sugar, and they are found in the cell. Okay, these are randomly distributed here, and whenever these ATGC link with the sugar and the phosphate, and they are referred to as deoxyribonucleotides. So let us know, these are the deoxyribonucleotides which are randomly occurred in the cell. Let us mark in this way. Suppose these are the nitrogenous bases linked with the sugar and the phosphate, called deoxyribonucleotides. Now these deoxyribonucleotides are the basic thing that makes a new strand of DNA. So what is the role of these deoxyribonucleotides here? They have to go to the chromosome, and they have to be joined with each other and form a long stretch of DNA. Suppose two deoxyribonucleotides join with each other, again another deoxyribonucleotide is joining, another. In this way, a long stretch of DNA will form. Okay. Here, to join this, to bring this deoxyribonucleotide from the cell to the chromosome, there is another enzyme which is very, very vital enzyme for the replication. That is called DNA dependent DNA polymerase. This is the main enzyme of replication. That's why I have put it in first number. And this is the main enzyme. So DNA polymerase will come and it will reach here. It will reach here. Suppose this is DNA polymerase. Okay. So DNA polymerase will start joining small deoxyribonucleotide here. Suppose this is one deoxyribonucleotide, and it will again bring another deoxyribonucleotide and join with this. Again another deoxyribonucleotide will come here, it will join with this, another. Ultimately here also the hydrogen bond established, and initially the bond was broken by helicase enzyme that will join with the new strand of DNA. Now see, this is the old strand and this is the new strand. These two new strand DNA helices are joining with each other, and the new DNA strand will have one old strand and the other new strand. So it will reach the end of the template DNA. So replication starts from this point. So always think about the new strand. So new strand will always start forming from the 5' end to the 3' end. So in the old strand, here is the 3' end and here is the 5' end, whereas in the new strand that will be opposite. If this is 3', then here will be 5', and new strand will form from 5' to 3'. So this process is going continuously. A strand is producing continuously, and this process is done by DNA polymerase, DNA dependent DNA polymerase. So DNA polymerase, it moves continuously. Now the new DNA has reached up to this point. Similarly, DNA polymerase has reached this point. Now if it moves further, helicase enzyme will break this bond and DNA polymerase will move further and strand will form continuously. It goes and till the end. Okay, this process is producing without any disturbance, and it is synthesizing continuously. That is why this process of synthesizing one strand from 5' to 3' is called continuous synthesis, and we call leading strand also. In the last class, I have shown you this continuous synthesis. Here, synthesis of DNA is continuously going. So this is also known as a leading strand, which is leading. And remember, the new strand of DNA always form from 5' to 3', and it is governed by DNA dependent DNA polymerase. So other enzymes are the helper only, like helicase, topoisomerase, they are the helper. Main enzyme is DNA dependent DNA polymerase. Okay, so let us come to the other strand. This is the old strand, but you know, in which the polarity is 3' to 5', we saw here, whereas in this case the polarity is just opposite. It is 5' to 3' is shown here. Here is 3' to 5', here will be 5' to 3'. Now always there is a mandatory, or there is a rule that DNA replication occur from 5' to 3', not from the 3' to 5'. So I am talking about the new strand, not for the old strand. So here this is new strand. It has synthesized from 5' to 3'. Here will be the same case. New strand of DNA will form from 5' to 3'. So for this, 5' will be this side and 3' will be this side. So how it happens? DNA has opened from this end. There is no space for any enzyme to move here. Actually it is very tight here. Initially the condition was same as this here also. Now for this, synthesis will be quite different. It is, the synthesis will be discontinuous one, and the small fragments will be produced like this. Let me show you one deoxyribonucleotide. This is one deoxyribonucleotide, and it synthesized from this end to this end. So this is the 5' end, this will be the 3' end. Now DNA polymerase enzyme starts polymerizing, bringing deoxyribonucleotide and joining. It starts from this point, not from here. Okay, this is DNA polymerase enzyme. is starting from this point and reach this point. So let me show you with the arrow. It has reached this point now. This became one fragment. Now let me rub this DNA polymerase. This is one fragment. Another fragment will come from the cell. That means one deoxyribonucleotide will come and it will polymerize in this way. Another fragment will come. So here polarity of this fragment is 5' is this side and 3' will be this side. Okay, another fragment is formed. Understood? Let me show with arrow. Now there is a gap here. Gap is created. I'll talk about gap later. Another deoxyribonucleotide has come and synthesizing of DNA starts from this point to this point. Again the polarity is 5' to 3', 5' to 3', 5' to 3'. Here some fragments are formed and these fragments are called Okazaki fragment after the name of scientist, and these strands are not synthesizing continuously, and this type of synthesis is called discontinuous synthesis. Discontinuous synthesis, and this is continuous synthesis, this is the discontinuous synthesis, and these are known as lagging strand. Lagging Lagging, lagging strands. Okay, now here is some different case will be here, as we have seen in continuous synthesis. This will not happen here. The case will be quite different. To synthesize these fragments, the process is quite different, as they need one primer, that means starter. Starter will be RNA. I am talking about this lagging strand. They are forming before forming these fragmented strands, some small fragment